Chapter 17 is the last in our unit of evolution. Now we'll talk about speciation, and that's the bottom line. If things are going to evolve, they're going to become new species. So we'll sort of define the changes that have to happen to become a new species. Like this picture, because colorful, beautiful. They're cichlids from a lake in Africa, and they're just representation of all the different species that might exist in one location. So a couple things about species. There we go. Again, a mantra you've heard all along. Before we could have molecular data, everything was classified on the way it looked. And some species look alike, some don't. So basically our best data now would be molecular data on how related things are to each other. But we can get by with some of the visual data and physical classifications. So this picture is just showing you that the same species might actually live in pretty different parts of our country, for example. And then even though these guys, they got some similarities, but they don't look perfectly alike. Wow, there's some real big difference between males and females. This happens a lot in the bird world. It's called sexual dimorphisms. Anyway, you also can have some species that really look an awful lot alike. Look at the spot pattern, the structures and stuff of these two frogs. Make them look like they're the same species, just some slightly different variations in color, but they're actually not. And they can't interbreed. And that's the bottom line of a species. It has to be able to interbreed and make viable offspring. Because if you can't perpetuate the species, what good is it? Now's a good time to point out the naming. So the naming system that we use for species came from Carl Linnaeus, early 1800s, where he basically classified things and gave them groupings. So kingdom, phylum, class, order, family, genus, and species. And right now here you see the genus and the species. The genus is always capital letter, species is always lowercase. So humans are Homo sapiens. This frog is Hyla chrysosceles. So how can new species arise? Well, we know that it has to be genetic, right? When you have a new gene, if that new gene gives you some fitness in the environment, allows you to reproduce better, you will keep that gene. This is just an example of how genes might change. So we start out looking at one species that has these two genes, A and B. And they're going to be designated lowercase just for clarity. So maybe they get these two species, this species gets separated by a geographical barrier somehow or another. And the A allele mutates just at this locus and becomes a capital A. Over time, that gets kept in the population. Now we have two capital A's. Notice it's a different color. Same thing happens down here. Maybe the B allele changes, and that gets kept in the population, and now it's a B. So these species may be compatible in all other ways, that they can mate and they can make an offspring. But it's entirely possible, in the scenario they set up in the book, that this allele is detrimental when it's in the presence of this allele. So if these two would mate, their offspring may have a lethal combination of genes. I can't give you an example of what that would be, but this is a scenario that these guys, Dozansky and Mueller, set up a while ago. Kind of goes along the line that genes change, and if genes change too much, you're going to have a totally different species. This is a diagram that's from the Campbell textbook, because it puts together a lot of the words and vocabulary that are highlighted in your textbook in one place. So I thought that you might like this as a little guide. So basically there's many different places that you could have a barrier between two species that they won't be able to reproduce anymore. You can have them before mating, you can have them after mating, and then you can have them in after the embryo develops. The bottom line is, for species to be the same, they have to be able to make viable, fertile offspring so that those can be perpetuated. So you can definitely have variation in the species, and humans and dogs are a perfect example of that. They still make viable, fertile offspring even if their variations are great. So what kind of barriers are we talking about? So pre-zygotic, or before you fertilize an embryo, habitat isolation. Two populations are separated, they never get to meet. good chance that their genes are going to change over time and they will become new species. Behavioral isolation, so maybe they listen to, there's going to be an example of frog calls. If the females don't like the sound of the males, they won't be attracted to them, you get no mating. Temporal isolation, they're active at different times of the day. Great example of that is flowers. There are some flowers that only open up at night. White ones tend to do that. There's a few of them. And they get pollinated by moths or bats. And if that same other similar flower is open during the day, you're just not going to be able to exchange pollen, and therefore no fertilization. So now that we can talk about, after we get over that part, we can talk about mating. There's physical, mechanical isolation. The structure of the reproductive parts just don't match. We can talk about that with flowers and pollen transfer. And then gametic isolation is a little bit more molecular. So maybe there are receptors that are not expressed appropriately, so the sperm of one species cannot bind the receptors on the egg of another species. And then after fertilization, things can fall apart. So sometimes the embryo won't develop. Sometimes the organism is born, like the donkey and the horse make a mule, but they cannot make functional gametes, so they can't make sperm and eggs, so they obviously can't reproduce. And then there's an occasional time that the hybrids can reproduce, and then their offspring have fertility problems, so later down things fall apart. Kind of all obvious things, but these are the different places to think about isolation. This is just a graph to show you that the more species are isolated, the better chance they're going to get genetic divergence, and basically it's a graph showing you that you increase in genetic divergence the further apart you are and the more time that you have been isolated. That's pretty obvious. Great example of this would be when the glaciers were in North America. So that big red dot, I think you can even see it in the small picture, shows the area that these fish lived in before the glaciers came down. And then the maximum glacial advance dipped into that area and separated their areas that they lived in, and they ultimately ended up into two totally different species groups, one in the eastern region, one in the western region. Not too surprising, come on with that idea. These are just the same pictures on. I'm going to skip through here. There's some island biodiversity ideas in this one. And this is the example of the Galapagos Islands. The Galapagos Islands are off the South America, and over time they got more and more isolated. And you always hear about Darwin's finches because he identified them in the Galapagos, and there are a lot of different finches, and you can see just in this gross picture from far away that their beaks are really different. Their beaks are different depending on the kinds of things they eat. They're seed eaters, bud eaters, and insect eaters. And these guys vary because the islands have such tremendous variation in their ecology. So, sorry about that. Dogs went crazy. Somebody was at the door. Anyway, back to Darwin's finches. So the Galapagos Islands are, let me zoom in just a smidge. They are a bunch of islands here, and each island has its very own ecosystem. So some places are dry, some places are wet, and the finches, because they are highly isolated on those islands, have developed into different beaks because they can eat different things. They're a perfect example of geographical isolation. This is called allopatric speciation because they are turning into different species in different areas. Sympatric speciation. Jump ahead to this picture here. Sympatric speciation is when things become different species or get isolated, but they're still in the same area. And an example of how that might happen is here with these frogs. They have behavioral isolation even though they're in the same area because they respond to different calls. The frequency is different in the calls, and you notice a greater variation in the call patterns of those green guys and the red guys when they're in the same area. Allopatric speciation is a variation in their calls, and you can see they're a little closer together when they're in different places because they aren't near each other. So there's less interaction anyway. So allopatric speciation is when you are geographically separate. Sympatric speciation is when you're in the same place. And these are just examples of how allopatric populations might be laying eggs at different times, and they're separated. Sympatric, they're in the same area. You'll oftentimes find sympatric speciation happening among plants because they can handle pollinating themselves. This picture is not showing you pollinating themselves. This is actually showing how some plants, flowers, have made themselves, have been selected to have parts that fit perfectly with certain kinds of pollinators. So here's a hummingbird getting and exchanging the pollen for that flower, and here's a moth sending his proboscis into that flower, and this flower has been designed or been selected for to have be pollinated by that moth. There's just pretty pictures. And then the last idea is that oftentimes you'll find species have a hybrid zone where there is a little bit of interspecies mating, and that means that that's a transition area. They may still interbreed a little bit, but as you get farther apart, the species are very different. And that's it. I would go back to this slide in the beginning just to use as a review. You may want to refer to that, all the different ways, barriers between species. That's it.